Thank you very much, and welcome to SciLife Lab from on my behalf as well. I'll try to give you a bit of an overview of the DDLS Data-Driven Life Science project, and then show you some examples of generating data and analyzing data from SciLife Lab in the precision medicine context. So let's see if I can get this going. Right. So the Data-Driven Life Science program has started a few years back. We think when we formed the program is that we are moving to very data-rich area in research. There is lots of data deposited in the life science domain, and we really need to change the way how we use this data in order to turn the wheel. We have been driving it experimentally, the field, but now we think that we can drive the development by data field. And the program is designed to harness the researchers in Sweden and harness the universities to do that. Really turn the wheel to the data-driven life science area. So what we have been we were very fortunate co-developing this program with Knut and Alice Wallenberg Foundation, and we were happy that we got the big 12 years commitment. All the major Swedish universities are involved. What we think is that we need to get talent in place first. So we have a very ambitious program of recruiting young investigators as PIs, 39. 20 are already recruited. We have also a PhD program and postdoctoral program with a significant numbers of young researchers. We have equally large program in Sweden on AI and robotics and autonomous systems on the data side called VASP. We have also bridging funding to connect the life science and computational science on the tech side together. So we're working together with DDLS and VASP projects, where we also have ethics and humanities part, where we consider the ethical aspects. Then we're also building capabilities on exactly around the question that was, what are we going to do? Where do we host the data? Do we have a capacity to analyze the data? How do we build this ecosystem working with the companies, which is also a still pretty much open question. So there is capabilities built up on the computational side. And all this we try to build in a unified program to an ecosystem where the fellows that we recruit and the researchers can work together, use the data resources, and work together also with the data providers at the SciLife Lab. So building this sort of ecosystem of data-driven life science here. And of course, partnering with European and worldwide initiatives, also partnering with industry is important aspect here. The precision medicine and diagnostics area, which I'm leading, and we have gathered national expert group from different domains to discuss monthly of the development of this. We have the fellows program and the PhD program, and we have also data science node, where we particularly look at the areas of the precision medicine and diagnostics, where we are currently building a data portal, which is going to be sort of a waterhole where you can see what data do we have in access, what data is produced, what things are happening in Sweden and in Europe and in world. And this is then, of course, connected with the data generation at the Science for Life laboratory. But what does this mean to individual patient? We are doing precision medicine, so I'll show you some examples of how to connect this. And as you know, the personalized medicine, we're trying to move from population and diagnosis-based medicine to actually individualize medicine. That means to see these colors of the patients before we start treating them, to match the particular treatments and therapy options. So what can digital tools do here? So here is a SciLife Lab developed digital tool, molecular tumor board portal, that we use in the Cancer Core Europe centers. That is a collaboration with 7 comprehensive cancer centers in Europe, across Europe. We all do same DNA sequencing. All the data is sent here. That data is harmonized, and the harmonized data is then compared with the 7 knowledge bases around the world to form individual report for each patient. And that way we can connect these world-leading cancer centers and provide them with an individual patient-centric report digitally. And this is how the report looks like. We're continuously developing this report based on the feedback. And this report is then discussed weekly in a European molecular tumor board. And that way we can really virtually connect the cutting-edge expertise in cancer round one patient in this individual report. And this is also a great opportunity to disseminate information. It's an e-learning situation where we can also develop the report. We can start working on what do we need to develop in the next generation. This is also an eye-opener, what we can do, what we can't do. Where can't we match the patients with cancer patients with the recent drugs? So this is data from our actual meta-analysis. So here we do sequencing of known cancer genes, and out of those known cancer genes, the variants that we see, 65%, are variants of unknown significance. We don't really know if they form as a biomarker to select the treatment. But there is even much bigger problem here. We can couple one drug to patient in addition based on this genome medicine. But the whole cancer community in the last 100 years have developed the science, and we know that there is multiple events that is turning normal cell into a metastatic cancer cell that kills the patient. And we are adding one drug. That's not going to work. We need to start adding cocktails of 3-4 targeted therapy drugs. So how do we do that? And this is something that we really need to move on. And this is where we think that the multimodal diagnostics, looking at the genotype, molecular phenotype, and clinical phenotype at once can do a difference. And I'll show you a few slides on actual science here. This is from ERA PerMed projects that I am PIing. It's an European collaboration, and I think these projects have been really good because they can put focus on certain areas and topics and connect front runners in this area. And I think and we have now disseminated this in a sort of national scheme. So what we did here is that we collected leukemia cases and did multimodal profiling. These samples are initially from Germany, that we did then lots of the profiling here. And if you look at the chronic leukemia cohort, the proteome analysis divides them into 6 different subtypes. But the real interesting data is when you connect these subtypes in the clinical data, and you can see that the proteome is very telling how is it going to go for the patients, very prognostic. So then we get a handle on the molecular phenotype and why is that so prognostic. The interesting stuff here is that the subtype 5 has the worst prognosis. And that was subtype that we could not define by any other means, not by genomics, transcriptomics, looking at the microscope. That was defined by proteomics. But because of the molecular phenotype, we can then start mining the actual biology here. And here we could see that the B-cell receptor signaling that is known to drive aggressive CLL is very low here. So we can define that there is an additional drivers. And then we use the proteomics data to find additional suggestions of drivers here. We can see defect splicing here and also develop molecular diagnostics to identify these patients. And with that molecular diagnostics, we could then go into additional cohorts around Europe and define this poor prognosis group. And that seems to be about 15 to 20% of CLL patients. And because of this molecular diagnostics, we can now direct these patients and try drugs on ex vivo setting and try new drug combinations and also initiate clinical trials based on this molecular diagnostics. There is also very interesting other phenomenon if we look at the other groups here. Two of the first groups have 3 copies of chromosome 12, which has been known since 80s, but we don't know why. Why is it beneficial for the cancer cells to have 3 copies of chromosome 12? But just by layering the genomics and proteomics data, we could go into the molecular details and realize that the B-cell receptor signaling is boosted by the chromosome 12 proteins. But depending on the lineage background of these cells, different proteins from the chromosome 12 boost the B-cell receptor signaling. So combining proteomics and genomics, we could re-stratify these patients, and then we could test that in a clinical trial setting, and B-cell receptor inhibitor actually wipes off the trisomy 12 effect. So just by layering this data, we can understand what are these complex genomics aberrations doing and how can we connect treatments to these phenomena. So that's showing the power of the multimodal profiling. So what is actually happening here is that the trisomy 12 boosts some proteins, but depending on the cell state, the synergic effect is different. In this case, in both cases, it's boosting B-cell receptor signaling, but by different proteins, by different molecules. So what I think we need to do is to start doing this multimodal profiling in order to do 2 important things. Here I use cancer as an example, but this is applicable for many other diseases, basically all diseases, where we try to understand genotype, molecular phenotype, clinical phenotype connections. To combine drugs in cancer and to find effective immunotherapies, the genomics alone is not going to solve this problem, because there is going to be a very complex regulation. We already know that we have epigenetic modifications, we have lots of somatic mutations and germline variants. They all form synergistic effects on the proteome that we treat. So if we would have extremely simplistic view of this, we have 3 molecular aberrations. These are the potential proteome combinations that you can form. But we know that we have hundreds or even thousands. Germline variants, somatic variants, epigenetic regulations. Not only in cancer, even in a monogenic diseases, the whole germline background is going to create this mess of molecular network. So how are we going to then direct treatment combinations on this? We need to start looking at the proteome that we also treat and connect that to the cellular phenotype and the molecular phenotype. We really need to start increasing our odds in this game by multimodal profiling. So what we need to do is that we need to think about the whole trajectory and how to analyze this data together, as an example that I showed in the CLL, that just by connecting two of these data levels, we can add and understand the data much better. So in order to do that, we have collected few local examples. We have a lung cancer project on the multimodal, we have a breast cancer project on the multimodal, and we have a leukemia project here. Maybe you heard some of these already. We have also added a national proteomics effort on this, connecting the expertise here in Stockholm, in Gothenburg and Lund, to actually add proteomics data to the genomics data, to work hand in hand with the Genome Medicine Sweden and the precision medicine centers and the comprehensive cancer centers. And then bringing these capabilities into clinical trials. So this is an example of a national trial that we are planning in cancer, where we want to connect these data layers in order to learn. And we think that the more that we do this analysis, the more we will learn. There is lots of low-hanging fruits. And I think this is urgent that Europe puts really effort on this. So just a few slides on the international outlook. In USA, we have a cancer proteomics effort that we are also well connected, but USA has put much more money on the multimodal profiling on this. There is a huge Chinese initiative on proteomics and proteome medicine. So 2 weeks ago, we had a human proteome organization meeting at the Dresden. So we collected the European front runners to discuss this. What do we do here? We really need to wake up here, and there is great examples. This is a few slides of my colleague Bernhard Kuster in Germany, that they're doing similar things that we are doing on a master trial, running tumor profiling on proteomics and phosphoproteomics in parallel with the genomics. So we had decided to team up with the German effort and the Swedish effort, so we can share experiences. Just one more slide from Bernhard showing that actually only very minor fraction of tyrosine kinase-driven tumors are manifested by the genomics. But if you look at the proteomics and phosphoproteomics, you can see that there is huge number of tumors that would benefit on tyrosine kinase inhibitors, but we just don't know the genomic determinants of those. So this type of data, we really need to form European initiatives and start working. So locally, we are trying to integrate this, but we need European and worldwide efforts on this. And I'll end my talk by showing this picture. It is actually Paris tube map with a peak hour traffic. I wanted to show you this picture because just by layering two separate data layers, you get much more information. Where are the bottlenecks, or where you should hit the drugs in the individual cancer tumors. So with that, I'm happy to take any questions.
Thank you, Janne. Wow. Questions? Otherwise, I have a question that, so thinking about yesterday, so we had Karin Liljelund, a patient that had received precision medicine and, it's under treatment continuously. So how would you say, how can we make this sort of accessible and also for the clinicians to sort of cope with that? You need to understand such layers of information. Do you see my question?
I think that's a great question, and I have two things that I think we should do. So first of all, I think that we need to understand that we can already today help individual patients. That's in an advanced molecular tumor board setting, where we actually do this multimodal profiling and can discuss in a very advanced discussion what drugs, what biomarkers should we use. And there, I think that the clinical decision support tools, like the molecular tumor board portal, needs to be developed to feed this complex data in different ways. But these people in these European molecular tumor boards and our local molecular tumor boards and our disease boards in the rare diseases that you heard yesterday, these people are very qualified to do very difficult decisions and complex data analysis. So that can be already done now, and that should be done in these advanced centers, and that should be catalyzed by fusing the research projects with the clinical routine. But that is not going to help large masses of cancer patients, because that's going to be only the late stage cases. So the next thing is that we need to migrate these lessons learned from molecular tumor boards into large-scale biomarker assays that we co-develop with industry and start testing. And then we need to put much more funding on developing biomarker assays that are high throughput, and then run those in a large clinical cohorts in a European setting. That way we can turn this cutting-edge medicine into a large population-based medicine in regional hospitals and in any university hospitals.
So many good messages to the European Commission. Thank you, Janne.